The next thing is, and here I'm going to say something that I think will hopefully shock your listeners who haven't heard this, because it shocked me. But if you say, okay, the next step is you take a sick person with measles, AIDS, chickenpox, COVID, whatever that is, any so-called viral disease, and you ask the question, is it possible to find a uniform, morphologically identical particle from any bodily fluid from that sick person? The answer is, if you look in the medical literature, there is not one case that that's true. Now, let me just tell you the people who agree with what I just said. Number one, 95 as of yesterday, health institutions have been asked, can you show me a study where a SARS-CoV-2, that's the virus, has been found in any bodily fluid of anybody with COVID? In writing, they say no. Then there's like the Pasteur Institute, the Robert Koch Institute, the CDC, all have said no. All the virologists who write papers saying isolation of SARS-CoV-2, if you ask them in writing, they say no. And here's another thing. Andy and I had the opportunity to present what we're talking about today to a group of lawyers and activists and doctors, and they invited a guy who was introduced to us as a laboratory pathologist and expert in laboratory virology in Wuhan, Chinese CDC for 20 years, and then a Yale pathologist for 20 years. And we asked him, is it possible to see a isolated example of SARS-CoV-2 from any person with COVID? And he said no. And we said, why? He said, because there's not enough to see. And of course, that begs the question of if there's not enough virus to see in an electron microscope, on what theory is it going to kill us all? But hang on. So Andy...
Trust me, it's invisible. Yes.
Andy had the presence of mind to say, what about if you took 10 people and mixed them together, their bronchial fluid? Would there then be enough virus to see? And he said no. And Andy asked him, what about 100 people? And he said no. And he asked him, what about 1,000 people? And he said, there's not enough to see. And then we asked him, what about 10,000 people? You mix all their fluid together, would there be enough to see? And he said no. And then he wouldn't answer anymore. So the reality is, everybody who knows anything about virology agrees that you cannot find this uniform, identical, morphologically identical particle in the bodily fluid of any sick person. Not with chickenpox, not measles, not mumps, not the flu, not HIV, not COVID. So the question is, why did they write 10,000 papers or so claiming isolation? And that's exactly what Andy just described. They take fluid, they mix it with bovine fetal serum and calf serum, monkey stuff. They spread it on vero cells, usually, or other types of cells. They poison it, they starve it, it breaks down, and they call that isolation, even though we now have two separate experiments that say, because the original experiment that did this by a guy named Enders, he actually did a control, and he did the whole tissue culture with nothing from anybody with measles, right? And he said, in quote, the result was indistinguishable. And when I read that, I thought, wait a minute, you just showed that the whole breakdown of the cell, which is what you're calling the virus, happens even if you don't add anything from anybody with measles.